Here we go. Here we go. Hello, everyone. So thank you very much for joining us today for the 8th ECI webinar of Translacore Consortium. So today, we are three people sharing this 8th webinar dedicated to ribosome translation and cancer. So I will do a brief introduction. So I'm Caroline Moyret, and I'm a professor at University Claude Bernard Lyon 1, and I'm doing research at the Cancer Research Center of Lyon in the team of Jean-Jacques Diaz, just like the two main speakers who are Virginie Marcel. So she's going to talk about RNA 2' O methylation, ribosomal RNA 2' O methylation in cancer. And also the second speaker is Sébastien Durand, also is going to speak about the 2' O methylation process of ribosomal RNA in glioblastoma. Virginie is more largely evoking the involvement of ribosomal RNA 2' O methylation in different type of cancer, more generally. So I will do a brief introduction, of our team, because we are working together in the team of Jean-Jacques Diaz. I guess everybody knows who is Jean-Jacques. And so the team here in the Cancer Research Center of Lyon is called Ribosome Translation and Cancer. So globally in the team, so we are studying alteration of ribosome composition, function, and protein production as well in different types of cancer, with two main aims. First, to get better understanding of the process by which ribosomes are involved in tumor development. And the second aspect, the second aim of the team, is to apply this knowledge to the identification of new biomarkers, and probably the development of new therapeutic strategies. As you may know, all the ribosomes alteration in cancer can be at levels of ribosomal RNA and ribosomal protein as well. So for ribosomal RNA population, polymorphism were identified in cancer, and also chemical modification. And so in the team, we really focus on some specific chemical modification of the ribosomal RNA. And also some researchers in the team have studied the role or the involvement of some ribosomal proteins in cancer, not really about mutation present in this protein, but more about the involvement on some isoforms, paralog, and also post-translational modifications. So as I mentioned previously, so in particular, Virginie and Sébastien are really focused on the methylation of ribosomal RNA, and some great publications were realized by the team now for more than 10 years, and in particular a cancer cell with Virginie Marcel. She will tell you about a little bit more later on. And so in Jean-Jacques teams, so we have different team projects and different team project leaders. So Marielle Barré is leading a project about therapeutic targeting of externalized proteins, so modification of proteins after translation that address to the membranes. They should not be addressed normally to the membranes. Philippe Bouvet is his project is focused on nucleolink function and targeting. Frédéric Cattet is leading a group about ribosomal RNA methylation involvement in translation and with some therapeutic targeting. Nicole Della Venadia has particular interest about modification of translation by some chemotherapy, so some chemotherapies resistance involved in translation, in particular after 5 FU treatments. So Sébastien Durand, sorry, will tell you a little bit more later, is particularly interested in translational regulation of cell plasticity, in particular in embryonic stem cells and glioblastoma. And Virginie Marcel, as I told you, so I'm belonging to the group of Virginie Marcel, so I'm working with Virginie. And so she's really interested in studying, and she's studied for years and years now, the ribosomal RNA methylation in different types of cancer and its clinical relevance. Sorry. So the resume of Virginie, who's going to speak just after me. So she has done her PhD here in Lyon at York. So York is the Cancer Research Center of the World Health Organization. I think it's the only cancer research center of the World Health Organization in the world. And she has discovered new p53 isoforms in cancer. And after she moved for a postdoc to Scotland, to University of Dundee in Jean-Christophe Bourdon team, she keep on working on some function of p53 isoforms, also p63 and p53 isoforms. And she joined Jean-Jacques Diaz group a little bit more than 10 years, and working on SNO RNA expression and ribosomal RNA 2' methylation in breast, brain, and lung cancer. She will tell you about that very soon. And so Sébastien, he has done his PhD in Montpellier. So Montpellier is south of Lyon in France. It's on the Mediterranean coast, in Jamal Tazi team, and in Institute of Molecular Genetics and Biology. So he was working on spliceosome, and after he moved to States, to Colorado, University of Colorado, first working on mRNA decay, and after in San Diego, and he arrived in Jean-Jacques team less than 10 years ago, and working on ribosomal RNA methylation in stem cells, in particular in brain. Is really interested in exploring brain cancer, but also breast tumors. So we will tell you very soon also. So now we are going to move to Virginie presentations. Thank you for your attention.
So thank you. Okay. So thank you for the kind introduction, Caroline. So I'm going to share my presentation. If I find it, it's here. Tac, tac, tac. Up. I think that you can see it. And up. Okay. So I said by Caroline, I'm going to share with you some data regarding the alteration of ribosomal RNA 2' methylation in cancer. So as a quick reminder, as Caroline said, we're working on ribosome heterogeneity, which has been shown at ribosomal protein level, but also at the level of the macromolecule, but also at the level of ribosomal RNA, which shows some chemical modification, but also nucleotidic variation. And so the current concept is that there is a different kind of ribosome exhibiting different composition can favor the translation of some specific mRNA and thus specialize the function of the ribosomes. So one example has been described by the group of Barna in California, where she shows that a ribosome containing RPL10A protein favors the translation of mRNA encoding extracellular matrix protein, whether ribosome containing RPS25 proteins favors the translation of mRNA encoding proteins involved in the P12 vitamin pathway. So as said in the lab, we're mainly interested by 2' ribose methylation. So in fact, the ribosomal RNA display different chemical modification. So in human, 112 sites have been described as 2' ribose methylation, meaning that methylation has been added to the ribose. More than 100 sites present some pseudo-redylation, and pseudo-redylation corresponds to the isomerization of the uridine. And 12 base modifications have been described, meaning acetylation or, for example, base methylation. So regarding 2' ribose methylation and pseudo-redylation, their precise localization within the ribosome is given by a non-coding RNA called SNO-RNA. So regarding the methylation, they are in fact guided by CD-box SNO-RNA. So this CD-box SNO-RNA interact in a second specific manner with the ribosomal RNA, thus allowing the enzyme, the methyltransferase fibrillary, to precisely catalyze the addition of methylation to a particular nucleotide, giving this particular localization of 2' methylation that you can see here. Regarding pseudo-redylation, it is guided by HACA box SNO-RNA, which this time, in fact, guides the enzyme, the discering, on the pseudo-redylation. So in the lab, we're mainly interested with 2' ribose methylation. And so our objective is first to determine what is the heterogeneity of 2' ribose methylation in real life, in particular in cancer. What is the role of this methylation on ribosome activity, their impact of translatome, and of course, how they are involved in cancer hallmarks. And so today I'm going to give you first a short historic overview of what we've done in the lab. So first, we observed in a cellular model where P53 was inactivated, that in this case, we've got an increase of fibrillary expression, so the methyltransferase, alteration of P53, and increased expression of fibrillary was associated with an alteration of methylation of ribosomal RNA, however, at some specific sites. And in addition to that, we observed that inactivation of P53 was associated with an increased translation of some particular mRNA containing a cis-regulatory element called internal ribosome entry site. So the problem with this first demonstration that RNA 2' methylation is altered in cell line is that in fact it was only an association between alteration of methylation and the putative role in translation. So that's why in the second study that have been mainly developed by Frédéric Atté, we use in vitro translation assay. So what we did, in fact, so in cell line, we performed a knockdown expression of fibrillary to experimentally alter the profile of RNA 2' methylation. The ribosomes were purified and they were introduced in an They were introduced in a tube with containing a ribosomeless rabbit reticulocyte supernatant, containing all the translational eukaryotic translational factor, but also tRNA and ATP required to perform the translation. And we also introduced in vitro synthesized mRNA containing either, I will say, a canonical 5' UTR such as globing and a 5' UTR containing an ROS structure, this one coming from the CRP viruses. And thus, in this condition, we observe that alteration of methylation has no impact on translation of mRNA containing a 5' UTR without structure, while it significantly decreases the translation of mRNA containing an RS, thus demonstrating that alteration of methylation impacts the translational activity of the ribosomes. At the time, the question was, but is RNA 2' methylation altered in cancers? And so the problem is that it was in 2012, we have no way to analyze RNA 2' methylation except using RT-qPCR on more than 1 microgram of RNA, so completely infeasible in tumor samples. So that's why we first analyze FBL expression in 3,000 breast cancer samples. And as you can observe here by immunohistochemistry, we identify that 10% of breast cancer tumors completely lack the expression of fibrillary compared to the others. So this was suggesting that alteration of expression of fibrillary was certainly associated with alteration of methylation in some cancers. So during this study, the team of Henrik Nielsel developed the RiboMedSec approach, which allows to analyze RNA2' methylation. So it's a method based on an RNA-seq approach, and in fact, the first step of RNA-seq is dedicated to analyze the presence of methylation. Indeed, the fragmentation of the total RNA is performed in presence of an hydrolyzed alkaline hydrolysis. And in fact, it has the property to hydrolyze the phosphatase bond between two nucleotides, except in presence of a methylation. So in this case, if you've got a presence of methylation on a particular nucleotide, this nucleotide will never be present at the 3' end of the reads, and the following nucleotide will never be present at the 5' end of the reads. And so by mapping the read on the ribosomal RNA, we can count the 5' and 3' end read, and thus determine the score of methylation compared to the overall nucleotide. And thus, what is a score of methylation, the C-score? A score of 0 means that no methylation is present at a specific site in all the RNA present in the sequencing, and comparing to a C-score of 1, meaning that all the ribosomal RNA present a methylation at the position of interest. And in the middle, a score between 0 and 1 means that we've got a mix of RNA showing a methylation or not at a particular position. So this RiboMedSec method has been developed for yeast. Yeah, after that, it has been done for human cell line. And in fact, in the lab, in collaboration with some platform of CRCL, one of sequencing and one of bioinformatics, we developed numerous methodology and bioinformatics tools to analyze large sample of cohorts. Because one of the problems when you analyze more than 20 samples is that you will have to perform several libraries, and thus you will have a bias due to the different batch of sequencing. And thus we develop some normalization to avoid this batch effect. So using these tools, we first analyze a series of breast tumors, of about 200 breast tumors. So these tumors have the particularity to represent all the different subtypes of breast tumors, including luminal tumors, which are dependent upon estrogen or progesterone receptor, but also triple negative breast tumors. And these tumors, in terms of transcriptomic, are largely different. So we analyze frozen biopsy coming from these different breast tumors, and thus we analyze the RNA2' methylation levels. Here you can see the C-score from 0 to 1 for all the different RNA2' methylation sites. And here, each point represents a breast cancer tumors. So first, you can observe that more than half of the site present a stable RNA2' methylation levels, even if the tumors are largely different from triple negative to luminal breast cancer. This implies that these methylation levels are very consistent within the tumors. And when we look at the localization of this site within the ribosomes, Bruno Klaus identifies that these stable RNA2' methylation sites are located within the functional domain of the ribosomes. It suggests that these sites are essential for the fine-tuning of the ribosome structure. So in contrast, we observe that about 30% of the sites show high heterogeneity between the tumor. As you can see, we have some tumor with no methylation at the site compared to other tumors which have almost 100% of methylation at the same time. So this site showing a huge variation in their RNA2' methylation levels are mainly located within the second layer, close to the functional domain of the ribosomes. So implying that this site, by the presence of absence of methylation, can help to finely shape or refine the functional domain regions of the ribosomes. And of course, there could be a source of ribosome plasticity. So to determine whether this site may have an association with some clinical data, so first we analyze correlation between RNA2' methylation levels that you can see here and different clinical characteristics of breast cancer, including subtypes and grade. So what we observe that we identify 4 sites for which RNA2' methylation level is significantly different between either breast cancer subtype or grade. For example, we observed a decrease of methylation at the site GM1447 of the 10S, 18S, rRNA in triple negative breast cancer compared to luminal tumors. In reverse, we have identified another site for which methylation level is increased. One of the problems when you analyze a tumor as the bulk samples is that you have an heterogeneity of the tumor, meaning that you have epithelial cancer cells, but also other kind of cancers, including cancer cells, including stroma, fibroblasts, or also immune cells. So to determine whether this alteration of methylation really rely on the epithelial cancer cells, we analyze in parallel a panel of cell lines representative of luminal breast cancer or triple negative breast cancer. So we analyze RNA2' methylation for all the sites. And once again, what we observe with this cell line is that we have some sites for which there is no variation of methylation. However, about 30% of the sites for which we have a huge heterogeneity in the methylation of level. If we look at the 4 sites that we identify in tumors as associated with clinical data, in fact, we still observe the large decrease of methylation level at the sites 18S 1147 in triple negative breast cancer cell line compared to luminal ones. And it was the reverse for the other site, suggesting that what we observe in tumor samples really come from the epithelial cancer cells. So in the lab, we're interested in breast cancer, and one of the major clinical issues in breast cancer is the progressive metastatic progression. And so this event relied on particular biological process called epithelial to mesenchymal transition. So in fact, the epithelial cells are now, thanks to alteration of expression of some transcription factors such as Z1, snail, and twist-N, to transdifferentiate within a mesenchymal cell line. So this cell presents higher motility and also invasive characteristic. And thus, expression transdifferentiation from epithelial to mesenchymal cells increase the metastatic progression of breast cancer patients. So what we analyze RNA2' methylation in a panel of cell lines exhibiting different mesenchymal or epithelial properties. And as you can see, only based on the RNA2' methylation profile, we can distinguish mesenchymal cell line in green from epithelial cell line in yellow. So to really understand what are the differences in terms of RNA2' methylation, we switch in another cellular models. So we use HMEC, human mammary epithelial cells, in which the anti-transcription factor Z1 have been overexpressed, inducing the mesenchymal acquisition of mesenchymal properties. So we use these two cell lines to analyze RNA to primer methylation by Ribosomal, and we analyze that in the total RNA samples, in the cytoplasmic RNA samples, but also in the RNA coming from the polysome, to be sure to analyze the RNA to primer methylation profile of ribosome, which are translationally active. And as you can see on this example, we observe alteration of methylation. And in particular, we observe a decrease of methylation at the site 2402 in cells overexpressing ZEB1. So this site was shown as decreased in total cytoplasmic, but also polysome ribosomal RNA. So to determine whether we've got an association between methylation level at this site and expression of the empty transcription factor ZEB1, we analyze a cohort of 71 triple negative breast cancer. Why triple negative breast cancer? Because we know that these breast cancer are enriched in mesenchymal properties. And we analyzed and we correlated the expression level of ZEB1 here compared to the methylation level at this particular site. And as you can see here, we observe an anti-correlation between expression of ZEB1 and 2' methylation at this particular site, suggesting a link between this EMT transcription factor and methylation at the site 2400. To understand why there is this particular change in methylation, we analyze expression level of different SNO RNA in ZEB1 overexpressing cells and epithelial cells. Also, we show no difference of expression of SNO in most of the SNO of interest. We identify two SNO RNA for which we observe a decreased expression in ZEB1 overexpressing cells compared to the other one. It has to be said that these two SNORD are in fact the guides for the site 2402. So the advantage of EMT, it's rely on the fact that it's a dynamic process. So in fact, the epithelial cells can, with the same, the same cell line and so the same genetic background, only the overexpression of one IMTTF is sufficient to induce mesenchymal acquisition of mesenchymal characteristic, and the removing of ZEB1 induce coming back to epithelial cells. So what we did that we use HMEC overexpressing ZEB1 under an inducible promotus, so allowing to come from epithelial to mesenchymal characteristic before going back to an epithelial one. And as you can see here, during this dynamic, we observe a decrease in methylation levels at site 2402, as observed when we just compare ZEB1 overexpressing cell to the other one. And when we go back from mesenchymal to epithelial cells, the methylation levels go back to normal basal levels, meaning that in fact, it's a dynamic variation of RNA to primer methylation during the change in cell identity. And what is quite interesting is that, in fact, it was completely parallel with a change in expression of the two SNO RNA, which guide the methylation in this particular site, with a decrease of the two SNO RNA and an increase of their expression to come back to something quite equivalent from epithelial cells. So thanks to an analysis of RNA to primer methylation by Ribosomal in breast cancer, we observe that in fact alteration of methylation is restricted to about 1/3 of the site, meaning that this site may have regulatory effect. This alteration is not random, since alteration of methylation at this particular site is associated with a specific tumor type, drive, but also aggressiveness. And in fact, by analyzing epithelial to mesenchymal transition, we also observe that variation of RNA to primer methylation is something that is dynamic, but also reversible. And now the objective is to understand what is the role of this methylation in EMT and thus in metastatic progression. So, of course, I would like to thank all our collaborators, including all the members of the Jean-Jacques Diaz team that have been involved in this project. And of course, thank you for your attention. So maybe we are going to move to Sébastien, and we are gonna to share the question in the same time, if it's okay for you. So now let's just place to Sébastien.
Okay. So, hi everyone. So, first of all, I would like to thank Caroline and Adolfo for this kind of invitation to present our work. So today I'm gonna tell you a little bit about I'm gonna share some recent and preliminary data we actually generated pretty recently on the alteration of 2' methylation of the ribosomal RNA in glioblastomas. So in the lab, indeed, we are pretty interested in high-grade gliomas, which are the most frequent primary brain cancer. So these tumors actually have a glial origin, and actually they remain mostly incurable despite a very strong and tough therapeutic procedure, which consists in surgical resection, radiotherapy, and combined chemotherapy. So there are three main subtypes of high-grade gliomas, which are the oligodendrogliomas, the astrocytomas, and the glioblastomas, which are basically stratified based on histomolecular characteristic. And for example, mutations on the IDH1 genes will separate the glioblastoma, which are IDH wild type, from the oligodendrogliomas and astrocytomas, which are IDH mutated. And then the oligodendrogliomas, they are further separated from astrocytomas based on the presence of a co-deletion of the chromosome 1p and 90q. So among those tumors, actually glioblastoma, they are the most severe, as for example, the median survival is about 15 months. And this is typically due to a very strong resistance to treatment and a systematic recurrence upon treatment. And we know that this is actually caused by a very strong cellular heterogeneity of these tumors. And this heterogeneity actually has been recently highlighted, I would say, by the lab of Mario Suva, where they show using single-cell RNA-seq that all the glioblastoma tumors are actually composed of distinct populations, which are present at different proportion. So you have, for example, the oligodendrocyte progenitor cell-like population and the neural progenitor cell-like, and the astrocyte cell-like population. So these three populations actually they really resemble to some normal population you can find in the brain during development and also in adult brain. And you also have this fourth population, which is called mesenchymal-like, which seems to not have any equivalent, I would say, in normal brain, but which is also very present in tumors. And so the relative enrichment of these different and distinct population will actually generate a bulk transcriptomic signature, which is actually commonly found in the literature, with tumor classified as mesenchymal when they are particularly rich in mesenchymal-like cells. Some other are classified as classical when they are composed of AC-like cells. And also you have a third subtypes, which is called proneural, when it is enriched in OPC-like and NPC-like cells. And in addition to this strong heterogeneity of glioblastoma tumors, we can also find in these tumors some cell population which display actually a very strong degree of plasticity and stemness. Some people, they call this kind of cells glioblastoma stem cells, but there is also some other cells which display a very strong plasticity. And we know that because of the presence of these cells, some subtypes, they can switch to one to another one upon some stresses, like, for example, hypoxia or low nutrients, but also upon treatment. And this is why you have a strong recurrence in glioblastoma. That's because upon treatment, some tumors, they can switch from one subtype to another one. And basically, in the lab of Jean-Jacques, we're trying to investigate whether translation regulation could actually be implicated in the establishment of specific cell identity in glioblastoma, and whether it could also contribute to the acquisition of plasticity and stemness properties by some cells, and whether by doing that, those transcriptional regulation, they could participate in the disease etiology. And as Virginie said, we're doing that by looking at a very specific angle, as we are mainly interested into the translation machinery, the ribosome, and trying to see whether this ribosome could be somehow contributing to those translational regulations. So I'm going to try to not be too much redundant with Virginie, but just an introduction on the ribosome. So this is a huge ribonucleoprotein complex, which is composed of two subunits, the large 60S and the small 40S subunit, that assemble together to give this 80S ribosome. So this ribosome is composed of 80 ribosomal protein and also composed of 4 ribosomal RNA. And I insist those RNA, they are really important because they are the one which are carrying the catalytic activity of the ribosome. And indeed, like so far, it's true that the ribosome has been always thought to be a very monolithic entity with a very stable composition, but we are now gathering evidence that actually the ribosome composition could display some degree of heterogeneity. And this is true at the protein level with, for example, some change in ribosomal protein stoichiometry, like the work of Maria Barna in stem cells. But you can also have like the presence of paralogs that will replace the canonical ribosomal protein. And this is actually even more true at the level of the ribosomal RNA with all the chemical modification that can occur, which consists, as Virginie said, in base modification, but mostly in pseudo-reudylation and ribose 2' methylation. And I think a big question in the field now, as Virginie said, is whether this heterogeneity could provide the ribosome with some specific activity that will make the ribosome a major actor of translational regulations. And this is the concept of the specialized ribosome. And in my group, but this is basically what we're trying to do is to try to test this hypothesis in the model of gliomas by being mainly focused on the chemical modification of the ribosomal RNA, which is also sometimes referred as a ribosomal RNA epitranscriptomics. So again, I think I'm going to be pretty fast to not be too redundant, but we are mainly focused on the 2' methylation of the ribosomal RNA, which consists in the addition of this methyl group at the 2' position of the ribose. So this modification is catalyzed by an enzyme called the fibruarin. And indeed, it is precisely guided to a specific nucleotide of the ribosomal RNA by this non-coding RNA from the family of the CD box non-RNAs. And as Virginie said, these non-RNAs they actually form a complex with a protein including the fibruarin, and they will precisely guide the fibruarin on the nucleotide to modify on the ribosomal RNA by a base pairing. So there are about 110 2' methylated sites which are known, and actually not randomly distributed in the ribosome, but instead it looks like they seem to cluster in functional centers. And we have now some evidence that a single 2' methylation could be very important for the activity of the ribosomes. For example, could be important for the interaction of the ribosome with what we call ribosome-associated factors. It could also be important for the activity or the selectivity of the ribosome, and also like some recent evidence that some modification, they could be also important for the ribosome biogenesis process. And this study of the 2' methylation has been recently facilitated, I would say, by the development of this technique called the RiboMedSeq by the group of Henrik Nielsen and Yuri Motorin. And this technique is really, really interesting because it allows actually the precise quantification of the methylation level at each single known position in one run of RNA-seq. And just to remind you, even Virginie described that very well, just to remind you the principle is based on the fact that the methylation on a nucleotide position will protect this nucleotide from hydrolysis. And that can be detected basically by doing RNA-seq and counting 5' read ends. And what you observe is like every time you have a nucleotide which is modified by a 2' methylation, you will never observe a fragment or RNA reads that start or finish at this position. And from this covering, we can imply a C-score which corresponds to the percentage of methylation. And as Virginie said, this percentage of methylation means that when it's 0, no ribosome will be modified at one specific position. When it's 1, it means that all the ribosome will be modified at one position. And when it's in between, it will mean that you have a mix of ribosome that will be modified and ribosome that will not be modified. And this is for us what we really consider as a heterogeneity of 2' methylation. And with the tools we develop at the CRCL for quantification of large cohort that Virginie told you about, we actually apply this technique to a cohort of high-grade gliomas, which were composed of oligodendrogliomas, astrocytomas, and glioblastomas. And the first observation was like exactly the same as what Virginie told you in cancer cells. We actually observe that about 75% of the sites are actually fully methylated with a C-score about 1. And only 25% of these sites show a very partial and heterogeneous methylation. So that was consistent with what we observe in cancer cells. But the main information is like, if you look at carefully, the glioblastoma, they are really separated from the other gliomas and even from the normal samples, showing that these glioblastomas have a very specific alteration of their 2' methylation profile. And the interesting point is, like, if you now consider only the sites which are heterogeneously methylated, they were actually sufficient to segregate the glioblastoma from the other subtypes of gliomas. And so this observation, basically, they were indicating that the glioblastoma seems to have a very specific alteration of the 2' methylation profile, and that this alteration relies mainly on sites which are heterogeneously methylated. So then we were interested to see, can we identify sites which is very specifically modifying 2' methylation in glioblastoma compared to the control non-tumoral samples? And indeed, we could identify a couple of sites. And this is very interesting because among the sites, some of them, it looks like they are located in some very important location to the ribosome, such as, for example, the sorcin-ricin loop, which is very important for ribosome elongation. And I will come back on that later. But we also have like some sites which are really close to the peptide exit tunnel. And we also have many sites which are differentially methylated in glioblastomas, which are present in this expansion segment that could have function in IRS mediated translation, or for example, translation initiation, sorry, and I will come back on that later. So when we got this result, actually one limitation of this is, like, as I told you, glioblastoma, they're actually really heterogeneous. So we're thinking, okay, can we find a way to try to narrow down this analysis to some specific population and try to get around this problem of glioma heterogeneity to see if we can identify specific population with specific alteration of the 2' methylation. And to do that, what we did was to establish a collaboration with Erika Kose here at the CRCL. So what Erika did, like, she used glioblastoma tumors with some specific transcriptomic signature, and she could derive like cell lines which are particularly enriched in 1 or 2 like glioblastoma population. So for example, she derived cell lines which are classical, which is mainly composed of like astrocyte-like cells. She could also derive some cell lines which are mainly enriched in mesenchymal cells, and also cell lines, which are specifically enriched in NPC and OPC-like cells. So we said maybe we can use this cell line to try to do a ribomet-seq analysis and see if we can narrow down alteration of 2' methylation to a specific cell population. And this is what we did. And indeed, we could identify sites which are differentially methylated in those 3 different populations. And actually, some sites show a very strong differential of 2' methylation, like this one. And actually selected among the sites, some of them which also are located in some interesting part of the ribosome. And so, for example, we found these 2 sites which are located in the 18S ribosomal RNA, so in the small subunit. So the AM576, and the GM867. And they're interested because, so first, there's a nice differential of 2' methylation, especially between classical and proneural. And very interesting because it looks like at the first sight, they are located in 2 different or 2 distant helix. So the expansion segment 6A and the helix 17. But actually, when you look at carefully, those 2 helix, they're actually clamping 2 ribosomal protein, the RPS9 and RPS24. And this structure, which is composed of these 2 helix and the 2 ribosomal protein, we know that they are really important for the recruitment of the initiation factor 3, via the 3J and the 3B subunits. So one speculation we could make is like these 2 methylation, when they change, they could maybe modulate the way this structure could recruit the initiation factor 3. So it could be maybe implicated in the translation initiation modulation. So another example, and Virginie also like told you a little bit about this site, that's the GM1447. And this is really interesting because this site has actually been identified by another team as playing a role in the stemness in the model of acute myeloid leukemia. And this site is actually located on the base of this expansion segment 9, sorry. And that is interesting for us too, because we know that this expansion segment interact with WAC1, which is very important, or at least implicated in the IRES-dependent translation. And indeed, this expansion segment 9 has been identified by the group of Maria Barda as being able to interact directly with some IRES-like structure in the gene coding for the OX protein. So this is very interesting that we have a site just in the middle of this IRES platform, I would say, which is modulated in the glioblastoma. And again, another site which is interesting, and Virginie also described this one, so it's a little bit redundant, but this GM4402 is also we say flashing in the glioblastoma because he has a strong differential. And this site for me is really interesting too, because it looks like it's located right next to the peptide exit tunnel on the surface of the ribosome, right at the place where the peptide is getting out of the ribosome. And this is important because we know that this part of the ribosome is a hub for interacting with protein modifying enzyme and also from chaperone, which are taking in charge the peptide which is being synthesized. So maybe like the modulation of the methylation at this site could somehow play a role in this process. So just to summarize, so we actually find that there is some specific 2' methylation alterations in the different population composing glioblastoma tumors. But something that is interesting is like those cell lines actually, they partially retain this capacity that the tumors have to switch from another one to another. So it means that in those tumor cell lines, we actually retain some cells which will have some plasticity and stemness capacity. So we are wondering, maybe we could also explore whether the 2' methylation could somehow be affected depending on the stemness and plasticity capacity of the cells. And for that, what we did for this each single cell line, we actually culture them in condition where we're enriching in cells with a high plasticity and stemness, and this is sometimes referred to glioblastoma stem cells, as I said before. And we're comparing that with cells which are more differentiated and which were actually deprived of any stemness and plasticity capacity. And when we did that, indeed, we again observed some interesting differences. And here, for example, it's in the context of the classical cell line. And we could also see some variations, some of them which are really modest, but some of us really strong in terms of differential of expression. But something that was interesting in this condition is that you have many sites which are clustering actually in some specific position. And I took this example of these 3 sites, which seems to cluster in this structure, which is right behind the peptide transfer center. And this structure is really interesting because it's composed of 2 stem loops which are interacting together through the loop. And we already know in the different model that modification of the methylation in this structure is really affecting the capacity of the ribosome to translate. And another clustering which is interesting, that's those 2 sites, because they're actually located at the base of the starcine-ricin loop. And we know that the starcine-ricin loop is actually really important for the ribosome function, as it interacts and stimulates the GTPase activity and regulates the activity of the elongation factor 2. And this is really important for translation and elongation. So again, maybe like variation of methylation on this position could somehow affect the way that the ribosome will elongate during translation. So just to finish, summarize what I told you. So we have some preliminary data actually that the 2' methylation is clearly affected and altered, I would say, in glioblastoma tumors in patients. And we have tried to narrow down this alteration to be more precise. And we actually found that it seems that this modification is also really dependent on the cell type which is composing those tumors, with differences in a different cell population of the glioblastoma tumors. But we already starting to find evidence that this methylation could also be affected and modulated during acquisition of plasticity and stemness. So right now we are currently testing, functionally testing whether the site and those candidate sites are very important for translational regulation. I'm not going to show you data, but instead I'm gonna show you what kind of tools and models we are developing for testing those hypotheses. And actually, in addition to the cell line we already have, we're starting to generate a bank of different model of glioblastoma tumor rates. And I'm not going to go too much into the detail, but what I want to just illustrate here is that when we develop those models, they actually stay very, very similar to the parental tumors. And for example, even if you test like histomolecular alteration, mutation, etc., they really keep all those alterations. And this is really interesting because we can keep those tumors for several weeks. They're still growing. They still retain the microenvironment. So this is a very powerful, I would say, model to study glioblastomas. And we could also do that for the other kind of gliomas. So here I just put an example of astrocytomas, where again, if you compare the characteristic of the tumors with the parental tumors, so with the organoids, you can see that you keep exactly the same characteristic, the same proliferation index, etc., etc. And with that, I would like to finish by thanking all the people which have been implicated in this work in the Jean-Jacques Diaz team, and also our collaborator for the help, and also the platform from the CRC that were engaged in this project. And I will be happy with Virginie to take your questions if you have any. So I'm going to stop sharing.
I have a quick question.
Yep.
These snow RNAs, yeah, so the ones that regulate the methylation places, yeah. Who regulate those? Are those constantly expressed, or they are regulated by the cell type or the state of the cell? Or if you put them in the cell, it will automatically methylate the place in the ribosome?
The answer, you can complement if so actually, I'm just going to talk about the CD box snow RNA. So most of the time, those of snow RNAs actually are present in the entrance of what we call host genes. Mm-hmm. So there is example where the snow RNA will actually follow the expression of the host gene. So sometimes it's really, really correlated. And we have some example in the lab where when you have condition where the host gene is gonna decrease in expression, the snow is actually doing the same. But there is also some evidence that you have a decorrelation sometimes. And that's probably because as the snow RNA is actually processed because it's an intron, it has to be processed by the splicing machinery, and there is some step of maturation. There is room for also regulation on all these different steps. So sometimes, even though the host gene is expressed at high level, you could have a decreased expression of the snow because it's not maturated, or you have some regulation happening at the maturation steps. So for the HACA, it's a little bit different because they have their own promoter for some of them. So I would say they could have a more specific regulation independent of over genes, etc. And most of the time, we also have example that if we want to try to regulate the expression of the snow by depleting their expression or increasing the expression, that's actually pretty easy. Every time we try to find a way to decrease expression of the snow, it works, I would say. But if you also express more snow RNA, you can see clearly the increased expression of the snow RNA. And most of the time, that will impact in the same way the methylation or the modification, which is linked to this node.
Thank you for that.
Just want to comment a little bit on that. It has to be said that for some site of methylation, only one snow RNA is necessary. But for other sites, you can have several snow RNA that get the methylation of the same site, meaning that, in fact, you have a kind of redundancy of expression of snow RNA to keep the methylation at a particular position, for example. And you have also some snow RNA that affect two methylation at two different sites, meaning that maybe also you have some combination of sites which are very important to be modulated in the same times.
Can I ask a question, please? So hi, Virginia and Sebastian. I was just wondering, following up on that last question, so for these sites that you see the differential methylation in the glioblastoma, sorry, I missed your talk, Virginia, so, but do you see the SNO RNAs changing in the same direction?
So yeah, I would say for some of them, not all of them, we haven't tested all of them. I think we have tested so far 5 or 6 of them. We actually see a strong correlation to the SNO RNA expression. So in some like subtypes, it will be high and you will be associated with a higher methylation, and some other it's like lower. So I will say for almost all of them we tested so far, there is a correlation.
Yep.
So in
So
breast cancer, we did exactly the same, but in fact, in this case, it was not straightforward. It means that for some site and some SNO, you have a strict correlation, but in other cases, there is no correlation between expression level of SNO RNA and the methylation level. But in this case, and maybe also in the case of glioblastoma, it's because we don't have take into account the fact that one SNO RNA can have multiple copy within the genome. And so maybe there is a correlation, but it's quite difficult to see as just seeing the level compare of expression compared to the methylation, and something else should be taken into account, such as their integration within the SNO-RNP complex, in fact.
Yeah, there's clearly something that we don't understand with some kind of licensing factors or something that are allowing access of specific SNO RNAs. But for the handful of sites that we have gone on and knocked out the SNO RNAs and done RiboSeq and all the rest of the functional experiments to show that the ribosome function has changed, then we always, for those sites, always saw like a strong correlation between the SNO RNA level and the methylation site. Yep. Thanks.
Thank you.
Thank you.
There is a question in the chat. I can say it loud. Very nice. I don't have a microphone. It's from Haichen Merthani, and he or she asked, what would be the best strategy to target 2O methylation in cancer cells?
Well, if we imagine we can deliver whatever we want in those cells specifically, I would say like, so using like antisense strategy might work. So we could deliver antisense RNA. We use ASO or LNA like chemistry. So you can basically like by just base pairing, you can create a destabilization of the SNO, and it will impact your methylation. But the problem I will say, if I want to be picky a little bit, is like we never know if the SNO has only a canonical function into methylation, or if it could have also like function in some other processes, because we have example like SNO, they are implicating methylation of the ribosomal RNA, but they could also have like a role in splicing. There is a recent paper showing that some SNO RNA, they're actually regulating the inclusion of some ribosomal protein into the ribosome. So there is probably like some other kind of role of the SNO RNA. So targeting the SNO is always a little bit dangerous. So what we sometimes discuss in the community of people working on that is whether instead of targeting the 2' methylation, you could target ribosome with this methylation. And that is, I don't think we are at the level where we do that, but we could imagine some sort of like chemistry where you could affect a ribosome because it has this methylation profile in cancer cells. So more targeting the activity of the ribosome carrying those methylation than the methylation itself. But I don't know if you want to add something, but I would say for now, it will be targeting SNO RNA will be the easiest with all this progress we made on the RNA technology to deliver to the cells, etc. That will be the easiest. But the best case scenario will be like the methylation is making the ribosome a little bit different. That will make it targetable by some molecules compared to some other ribosome that have a different profile. I don't know if I answered the question, but I hope so.
There is a question by Elena Martins. So Elena, please go ahead.
Yes, hi, thank you very much. I'm sorry, my camera is not working properly. Follow up on the last question again. We have tried LNA here with our SNO RNAs, and actually I never managed to get LNA approach working. So is that working in your heads? For SI worked much better for us in CRISPR-Cas, but LNA we never got actually really good results.
Well, so I will let Virginie answer because I know you use LNA. Right?
So we use LNA, but in fact it really depends on the cell lines. So in fact, we design LNA against one particular SNO RNA, and we use the LNA in different panel of cell lines in lung cancer, breast cancer, ovarian cancer, and so on. And the way we use it is that we use it by genosis. You just put the LNA within the medium and to see whether it enters the cells. And so just by RT-qPCR to determine the impact of this LNA on the expression of SNORD, in fact, it was completely different from one cell line to another one, meaning that maybe the LNA chemistry is more prone to enter to some cell line compared to other one. I don't know really why or what could explain that. Maybe a change of some pore within the membrane and so on. And some cells are more prone than others to take LNA. So that's why also we switch to another technology, ASO, antisense oligonucleotide, and that we transfect within the cell, so that in fact, in this case, we have less heterogeneity in the way the cell take the antisense gapmer.
Yeah, okay. I think that's more or less our experience as well. Just for some reason, LNA doesn't work in prostate cancer cell lines. Also does, and so does SI, but also is a rather expensive way to go for.
Yes, of course.
Okay, thank you. Thank you very much. Nice stories.
I don't think there is any more questions. So, Caroline, you want to conclude or?
No. There are any other questions? So I wanted to complete the answer of Sebastian and Virginie about targeting probably. So also indirectly, we can check which proteins are specifically translated from this methylated, specific cancer methylated ribosome and probably target these specific proteins because they will be the results of this modification also. It could be also an hypothesis or a way to target this specific methylated ribosomes. So somebody want to? No other questions? No, I don't see. Adolfo, you don't see other questions from the panel?
I have one more last comment. I mean, I think doing cryo-EM studies on cells where specific SNO RNAs have been knocked out to look for structural differences in the ribosome is going to be really important in this field, and that might, give some clue about what kind of chemistry, molecules and things will, and I'm sure it'll actually be different from methylation sites, SNO RNA to methylation site, because depending on where the methylation site is, it can have really different effects on the ribosome structure and maybe only be important in specific stages of the elongation cycle, or who knows.
Yeah, I mean, you're absolutely right. I think like this kind of analysis give you really a hint about what will be the impact on the real feature of the ribosome, because for now it's only speculations based on what we think. But I think can be able to prove that your EM will be something important. The problem is it's also the model, like when you see how much material you need to do cryo EM sometimes. I know that so far right now with glioblastoma models it would be impossible to do it. But with the improvement of model, maybe we'll be at some point able to generate a lot of material for doing such an analysis in this model. But right now it will be really difficult.
Yeah, well, we're doing it on some cell lines where you can get enough material. So hopefully we'll see something and let you know.
Yeah, but I think it's a good idea. Sometimes you can, because those sites were methylated in all different cell line, so it will not be as physiologic than using the original system you're using. But maybe you can deplete these sites in a different cell line to see how it will impact the structure. Still give you some hints, So
yeah,
it's a good idea.
Cool.
Thank you.
So thank you. Thank you very much for all to join this 8th ECI webinar. And Adolfo, if you want to conclude.
No, as usual, I'm just happy to help here.
Okay, so thank you very much. Thank you.
Thank you.
Thank you so much.
Thank you for such a nice talks. So thanks, Sebastian and Virginie.
Thank you.
Thanks.
Thank you. Bye.
Bye-bye.